Hello everyone, I am Anunay Kumar Gupta. I have done MSc Botany from Allahabad University and currently I am pursuing MTech in Biotechnology from IIT Guwahati. Today in this video I am going to talk about on topic cytoskeletons, centrosomes and centrioles. When we talk about this topic, we need to first discuss what is a cell. So cell is the structural and functional unit of every living organism. When we see a cell, it has different organelles in them. So a question arises, these organelles in the cells, are they fixed or just freely moving around? So the answer to this is the cells are anchored through a protein network called as cytoskeleton. We say that protein network as cytoskeleton. Cytoskeleton mainly comprises of three things, namely microtubules, microfilaments, and intermediate filaments. When we see microtubules, they are made up of tubulin protein. Microfilaments are made up of actin proteins, and intermediate filaments are made up of different type of proteins such as keratin, desmin, etc. depending upon the location. In this video, we are going to focus upon microtubules which are made up of tubulin dimer, namely alpha tubulin and beta tubulin. Alpha tubulin and beta tubulin together forms a dimer giving rise to microtubules. So we are going to focus upon microtubules because our centrosomes and centrioles are made up of microtubules. So if we see a diagram of a cell, here we can see nucleus. Near the nucleus, there is a dense and dark structure which is called as centrosomes. When we see the enlarged view of this structure, we see that these structures are rod-shaped and hollow cylindrical, and they are surrounded by a material called as pericentriolar material. This hollow cylindrical shaped structure are centrioles, and this material surrounding the centriole is pericentriolar material, PCM. Together, centrioles along with pericentriolar material, we say it as centrosomes. Now, when we talk about centrioles, centrioles are only present in animal cells and in some lower plants. It's absent in higher plants. So, when we talk about centrioles, functions as its function is to form spindle fibers during cell division, and also it gives rise to basal bodies which ultimately give rise to cilia and flagella. So we say that centrioles are absent in plants. So how do plants form spindle fiber for cell division? In plants we have different structure called as mTOC. That is microtubules organizing center, through which plants form the microtubules which are necessary for formation of spindle fiber. Now when we see a cross-section view of a centriole, here we see a cross-section view of centrioles. We see centrioles are arranged in a 9+0 arrangement. Why I am calling this 9+0 arrangement? We will discuss it further. Here we see centrioles are made up of 9 triplets, peripheral triplets. They are present at periphery. These peripheral triplets are made up of microtubules, and they are namely named as A, B and C, with a central hub. This is central hub made up of protein, not microtubules. Every peripheral triplets are named A, B, C, and we see a connection between A and C. This is called as AC linker. While these peripheral triplets are connected to the central hub through radial spokes. Radial spokes. We see this structure as it is looking a cartwheel. So we also call the structure is also called as a cartwheel structure. And array is 9 plus 0 because 9 triplets are present at the periphery and in center no microtubules are present. So it is called as 9 plus 0. At the center no microtubules are present. When we see the formation of spindle fiber originate from these structures only, from the microtubules at the periphery. So for this video, we are going to finish this video at this moment, and that is all for this video. Thank you.